Hey, RP Plus / RPU, welcome back. Dr. James Hoffmann here, and today we're going to be going over some basics of physiology. We at RP know that sometimes you guys aren't always up to speed on a lot of some of the basic kind of exercise and physiology and biology and chemistry topics that we're talking about, and we want to make sure that you have a nice whole complete understanding on some foundational materials before you go on and start tackling the tougher stuff. So the goal for today's lecture is just trying to get you familiar with some of the basic terms in cell biology and chemistry that we use a lot when we go into more deep discussions later on. So for those of you who have taken biology and chemistry at the college level, you might be able to kind of glaze over this one. If you have not, you haven't taken bio or chem maybe since like high school days, this might be a good one as a quick refresher before moving on to some of the more complicated courses like exercise physiology and then any of the specialized areas for like training and dieting and such. So let's go ahead and get started. The first slide basically shows us kind of what we're going to be talking about today. First thing we're going to talk about are just kind of some foundational biological molecules that we need to be familiar with. Now, we can get into really crazy discussions on some of these things, but we're going to keep it pretty simple and pretty straightforward for the most part. So hopefully we won't diverge in any areas that are too deep. The next one we'll talk about are just some biochemical reactions and how they work in the body. And then last but not least, we'll go over some basics of cell physiology and kind of the bits and pieces of the cell and what they do and how they work. So why don't we go ahead and get started. So to understand a lot of concepts in exercise physiology, we have to have an understanding of matter. Now, we know if you take physics, we can start breaking down matter into really small subatomic particles. But from biochemistry and a physiology standpoint, we are primarily concerned with our functional unit, which is the elemental level, right? So all biological matter is composed of different combinations of elements. We generally, on our level as exercise physiologists, tend not to go down much lower than the element. So an element is just a substance that is made up of protons, neutrons, and electrons. No surprise there. We know that elements can exist kind of as single entities by themselves, or in the body, more often than not, they are bounded together in groups that we like to call molecules, right? So a molecule is simply just a group of two or more elements bounded together through chemical bonds, right? Now this can be the same element, like two hydrogen ions. It can be different things, like two hydrogen ions and a water molecule, right? So there's all sorts of different combinations that we find in the body. The base level that we're going to be generally dealing with are elements, and then we're going to see molecules kind of branch off of that and move into macromolecules. Now, if you need a refresher on elements, you can check out the periodic table. We're not going to go into that because that's something that you can readily look up on Google. No big deal there. So let's move on to the next one. We know that chemical bonds in molecules is where we find the predominance of the stored energy of that compound. And we know that chemical bonds can come in a variety of different forms. And here on the slide, you can just see some examples of that. Some of the big ones that we see more often than not are usually things like ionic bonds, which are bonds based on the electronegativity differences between elements or ions, right? So we have kind of an electromagnetic force keeping them together. We can see things like covalent bonds, which is where we actually see sharing of the valence electrons between two elements, right? So we can actually see their start to share electron orbitals, which is really neat. And we can see some other examples very similar, like hydrogen bonding, which again is an electronegative bond. And then even within that, we can actually see bonds occurring on multiple layers, right? So we can see just single bonds where we have kind of one shell and a covalent bond being shared. We can actually see double and triple bonds, which is a much stronger bond where more electrons are being shared. All sorts of different combinations of those things. And this is just kind of the short version. If you ever take a physics or a physical chemistry class, you'll find that there are lots more different examples of different types of chemical bonds. These are just kind of a few of the big ones that we see most commonly. Now again, one thing to keep in mind here, the energy from molecules generally comes from these chemical bonds, right? And when we break them apart, we tend to release that free energy back into the universe. Now this can just be pure entropy, pure heat, or within the body, what we're most concerned with is can we actually convert that chemical energy into a usable form, hopefully the energy of movement, right? Which we have to break down and convert multiple times over so that we can generate tension in the muscle and then translate that into the energy of movement, right? So let's go on to the next one. In the human body, we know that most elements do not kind of sit in their base form, right? Typically, the body is what we like to call an aqueous solution. The body's made of mostly water, right? And accordingly, what we see is when elements are placed in an aqueous solution, whether it's just dumping them in some water or the way that they're found in the body, they typically ionize, which means they start to have an electronegative charge. This is usually as a result of an imbalance between the number of protons and electrons that that element is carrying. And so we can actually see some examples in the body. If you look at the slide, really common ones that we find are things like calcium, which you can see has a 2 positive charge, potassium, sodium, chloride, and then down there we have some inorganic phosphate, right? These are really common examples of ions that we see in the body. Now, why is this important? Well, this difference in electronegativity makes them highly reactive to other things, especially things that have polar opposite charges. So the reason why we see this a lot in the body is because sometimes people will say, oh, calcium, potassium, these things have a net neutral charge. Well, as they exist in the free world, that is true. As they exist generally in your body, they tend to ionize, and we can actually see the protons or electrons become imbalanced to some degree, right? And this can happen through oxidation and reduction reactions, which is maybe a little bit outside of the scope of today's discussion. So just keep in mind, you can see organic compounds, inorganic compounds, and typically when we talk about them in the body, they tend to ionize, or meaning change their charge a little bit from either positive or negative. So we usually don't see them in their neutral form. Now, when they bond together with other elements and form molecules, typically we see this tends to balance out quite a bit. Not always, but more often than not, they tend to take on a neutral charge once they have bounded together with other molecules. Okay, makes sense so far? Nothing too crazy, I hope? Let's go on to the next one. Okay, so we are mostly concerned in our discussion with biomolecules, right? So we can look at inorganic molecules, and I've actually taken more inorganic chemistry classes than I'd care to admit. However, when we're talking about exercise physiology, we're primarily concerned with biomolecules, and of particular importance are big three. Hopefully no big surprise here. Usually we're going to be talking about carbohydrates, fats, we'll call lipids, and proteins, which are composed of amino acids. So we have some examples of what some of these look like in their chemistry format on the slide here. We can see the top one is an example of a carbohydrate. We have some examples of fats. We can see the really long carbon chain. And then that bottom one is an example of what a base amino acid might look like. And what we can see here is we have an alpha carbon with an amine group, which is that NH2 group on the bottom left. And then usually we have some type of carboxyl group on the end. And you can actually see on the top, the one that's labeled R, that is usually any number of different combinations of things which make the amino acids kind of unique. So there's not kind of a set piece on that R chain side group that you might see there. They are different from amino acid to amino acid. Once those amino acids bound together into different forms, they make proteins, right? Hopefully no big surprise there. This can take a whole bunch of different conformational shapes. Sometimes you can see an alpha helical structure, which is kind of like what our DNA looks like. You can see beta sheet structures, which is kind of like a flat membrane type structure. All sorts of really cool stuff. But for our purposes, we're primarily concerned with how elements form molecules and then lead to things like carbohydrates, lipids, and proteins, our big kind of three macromolecules. Make sense? And hopefully you guys, if you're already if you're following RP, you've probably heard of some of this stuff before. Hopefully no big surprise. But now we're actually going to see how these things play a role in the body. So let's go on to the next one. So carbohydrates, hopefully no surprise, is our most fluid kind of usable energy storage form. We say it's fluid because we can actually, it's not stored in a long-term sense, it's actually stored in a more short-term usable sense, meaning it's not hard to get to. It doesn't have to go through a lot of conformational changes to use it. And it's readily available during things like exercise. We store it in the muscle itself. And so when we actually start working that muscle tissue, that explicit muscle tissue, like your biceps, if you're doing bicep curls, well, guess what? There's actually glycogen stored right then and there to be used so you can do high intensity and or voluminous exercise. We also store some in the liver that we can release into the bloodstream to help maintain blood glucose levels, which can be drawn into the muscle for storage or for use as well. Now, we also know that carbohydrates are not limited to that. They can also provide some structural components in things like chitin and cellulose and in plant cells. In human cells, we don't really have any of that going on, so we don't have to worry about that as much. But one other thing that is particularly important for carbohydrates from a biochemistry standpoint is very often they serve as kind of a carbon skeleton for a lot of other very important biomolecules. Of importance that we kind of want to note are things like DNA, of course, and ATP. So the carbohydrate not only is something that we use to provide ourselves with energy, but it's also kind of a carbon skeleton for a lot of other macromolecules. Again, DNA being a pretty big one there, as well as ATP. So really important. We also know that lipids are important. Now lipids, we'll kind of call /fats, are more of a long-term energy storage molecule. They're in the human body They are much more difficult to get to use for energy purposes during exercise. Now, during resting conditions, normal conditions, we use them quite a bit, but they just logistically have a really, really hard time making their way into the cell to be used for energy production during exercise because of the number of steps involved. It just takes a really, really, really long time. So it's more of a long-term energy storage. And what we see here is examples of things like fat tissue, adipose tissue, right? So we tend to accumulate, if you're a male, tend to accumulate it more in the gut. If you're a female, it might be more gynoid, like in the hips and in the legs, something like that. That's 100% normal. Now, that's the kind of fats that most people are used to talking about, but one thing that's important to note about fats and lipids is they actually provide membranes for a lot of different biological structures. So the membrane is really, really important for the cell because it's something that can help create a barrier between the cell and other things. It can also have hydrophobic and hydrophilic ends, which means one end is kind of averse to water, one end is more accepting of water, which plays a big role in how it regulates things moving in and out of the cell. So most of the membranes that we have, things that kind of enclose cells or keep things in and out, generally come from lipid bilayers or lipid membranes, which are composed of fats. No surprise there. So not only for energy, but also a lot of our structural cellular structure is based on lipids as well. Last, and certainly not least, is the one most people are most excited to talk about, proteins. Proteins are long chains of amino acids, right? They provide a lot of different structures in the body. Obviously, the ones we're most familiar with are things like skeletal muscle, smooth muscle, cardiac muscle, no brainer there, but also all sorts of different collagen fibers are also made of proteins, no big surprise. A couple other things that people generally maybe aren't as familiar with that are worth talking about. They're also involved in enzyme production. So enzymes are really, really handy proteins that are involved in chemical reactions, and we're going to talk about those in a little bit. We also know they are a big part of cellular signaling and regulation. So proteins actually act as cellular regulators for almost all of our major biological and physiological processes. They kind of act as messengers or kind of like a mail courier that drops off a message to a cell. Cell picks up on the message and says, oop, I need to change in some way. And that activates a whole cascade of different biological systems that start initiating some of those cellular changes. Really, really neat. Last but not least, one thing that's important is protein actually kind of can act as what I like to call the hard drive of the cell. So our DNA, right, is actually contained in protein strands in the cell. It actually contains all the information about what makes you, you. It actually contains all the information about how you change over time as well. Really, really neat and interesting stuff. So most of our cellular processes are regulated by proteins, which is really, really noteworthy. So we usually just think of protein, biceps, right? Well, yeah, that's part of it, but actually most of your body is regulated by protein synthesis to some degree. So very, very important to consider proteins not only for their structural purpose, right, and like skeletal muscle, hair, collagen, fingernails, things like that. We also want to consider enzyme production, cellular signaling, and they contain all the information that makes you, you, which is very unique for this particular biomolecule. All right. Let's go on to the next one. One of the things that we deal with a lot in exercise physiology is a term that we like to call bioenergetics, right? Bioenergetics is kind of the sum and total processes of converting things into usable forms and or synthesizing new things as storage forms. So we can kind of break that into a couple different terms. First is metabolism, which is the total of all cellular reactions within the body. Now, metabolism can kind of be further broken down into two major subcategories. The first one, anabolism, which hopefully most of us are familiar with, which is the synthesis of molecules, the buildup of structures, potentially for use, for repair, or even for storage, right? The next one is catabolism, which is the breakdown of molecules. Now, this is something that I think is noteworthy because a lot of people just think of metabolism as this kind of process of breaking things down. But really, when we're talking about metabolism, we're talking about kind of the sum net effects of both anabolism and catabolism. So when we're talking about anabolism, the question is, are we building things up? If we're talking about catabolism, the question becomes, are we breaking things down? And this is something that's kind of a noob mistake that a lot of people will make, where they'll say, hey, this thing is anabolic, right? And it's something that inherently breaks things down, right? So it cannot be anabolic by definition. Anabolic is something that causes structures to be synthesized, built up for use or for storage. Whereas catabolism, we're breaking things down typically for energy production. Not always, but typically. Now, We know that we typically are talking about bioenergetics and metabolism in the context of eating food and doing exercise, right? So food and different nutrients serve three major purposes for us. They help promote growth and development of the body, whether that's actually during maturation or it's for skeletal muscle protein synthesis or tissue repair, all sorts of things like that. It actually provides us with a usable form of energy. So eventually we're going to have to break down that food and convert it into a usable form of energy. There's potential energy stored there. It needs to be transferred into a usable form of mechanical energy, hopefully, for us to move around and ambulate and do all sorts of fun exercise stuff. And they also help regulate metabolism by governing which things are available, how much storage of certain things do we have, how are we going to change under different conditions. So really, really useful. Let's go on to the next one. In our context, when we're talking about exercise physiology and bioenergetics, this process of converting foodstuffs into usable form, typically what we're trying to figure out is, can I take some metabolically derived energy and use it to generate muscle tension, right? Get our muscles to contract and then perform some amount of work or various exercise-related tasks. So I think this is kind of a step that a lot of people seem to forget and kind of miss. They usually think of like, eat carbs, hit PR on squat, right? Well, really there's a whole lot of things in between there, right? We are eating food to break those chemical bonds, release that free energy, but hopefully convert it into a usable form. The way that we do that is by generating muscle tension. So the energy that we consume from foodstuffs is used to generate tension in the muscle, which is used to do exercise, sport, and various other tasks, right? So we know that the presence or absence in some cases of nutrients will have a significant effect on your ability to perform well, whether it's a sport performance context or an exercise performance context. They'll have a direct effect on your ability to recover from exercise. They'll have a big effect on how your metabolism is regulated, and certainly will have a big effect on your body composition, right? So we want to make sure that we're considering not just foodstuffs as, okay, if I eat carbs at this time, I get this effect, right? We're actually having a huge chain of events of breaking things down and then converting energy into usable forms. So let's go on to the next one. So in order to transfer the foodstuffs into a biologically usable form, they must undergo a series of chemical reactions, right? Now, a lot of people don't like chemistry or don't take a lot of chemistry, but there's a couple big points that we have to consider. So when we're dealing with biochemical reactions, there's a couple kind of different subcategories that we can talk about. The first one you'll see on the slide are endergonic reactions. And these are reactions that do not necessarily happen spontaneously. I don't add chemical A to chemical B and I get poof, chemical C. These are examples where I must add energy into this system of these two reactants in order to get the products that I want. And the products can be any number of things, right? But in order for this reaction to actually take place, I have to invest energy into the reaction first, right? Whereas on the other hand, we can have what we call exergonic reactions, which is where we have chemical A and chemical B, and they can react fairly spontaneously. Maybe not perfectly spontaneously, but they kind of do their own thing. You don't have to encourage them too much. And as a result, energy will be released as part of the process. Another burp on the way. Twofer, really good. So in exergonic reactions, typically what we see is energy is released as a result of the reactants. So as the products come out, energy is released. Whereas the endergonic reactions, we actually had to put energy into the system just to get them to react and create the products that we wanted. Now, most of the reactions in the body operate on a level that I like to call coupled reactions. Coupled reactions basically say we have multiple reactions that are linked in multiple ways. One of the first ways that they're linked is through their products and reactants, meaning I had two reactants, they formed a product, and now that product is actually being used as a reactant for another biochemical reaction, and then the chain continues. So we can kind of see a recycling of products and reactants as we go through the body. And then one other thing that we can consider for coupled reactions is actually the energy cost and energy generated from many different reactions. So what we find is, in many cases, the energy generated from an exergonic reaction might drive the energy required for a completely different and unrelated endergonic reaction, right? And we can see multiple examples of this in the body. So with coupled reactions, we're basically dealing with how the body kind of recycles products and reactants, as well as tries to maintain some level of energy balance within the body. So let's go on to the next slide and we'll actually see kind of an example of this. So here on the top left, you can see an example of an exergonic reaction, right? The products have less energy than the reactants because they released energy as part of the reaction. So we can see the products generally have less energy content than the reactants. On the other hand, we have those energonic reactions where we had some reactants, they didn't really necessarily act on their own, we had to put energy into it, and then as a result, we got our product, and we can see the energy level of the products is slightly higher than that of the reactants. Now, we can see all sorts of examples of this in the body. And if you look at the bottom one, we see an example of what a coupled reaction might look like. So we can see here we have a glucose molecule that's being broken down into carbon dioxide, water, and heat being released. And we can see that the ATP being used or generated from that reaction is being used for something completely unrelated, actually generating a polypeptide bond from multiple amino acids. So we had some amino acids, we wanted to bond them together, it required energy. The energy released from this glucose reaction actually helped drive the creation of this protein. Really, really neat. One thing that is noteworthy on this chart that you should take note of here is, as you can see that red arrow going down, right? What does that red arrow say? Well, it says net exergonic reaction. So one of the things that we see in the body is that unfortunately, energy is not completely conserved or balanced throughout these processes. We know the body is not 100% efficient. Energy is lost along the way. And typically what we see more often than not is that energy is just simply lost as heat. And you can actually see that in the first part of the reaction where glucose is broken down into carbon dioxide, water, and then heat is released. So not every reaction within the body is 100% efficient, meaning there is entropy, energy lost to the universe along the way, and that is 100% totally normal. Our body's not 100% efficient. I think it's on point with that of like a light bulb or a diesel engine, so not super great in terms of energy efficiency, but not too shabby either. All right, so let's go on to the next one. Now, we just talked about these different chemical processes, sometimes requiring energy, sometimes releasing energy. Most of them do require some sufficient amount of energy to proceed. Now, they could have enough energy at rest and react spontaneously. Other times they might need a little nudge, a little poke, right? And this little kind of energy deficit is called the energy of activation for this chemical reaction, meaning this will not happen unless I put X amount of energy into this reaction. That is the activation energy the energy of activation for that reaction. Now, one of the ways that we can kind of make this burden of energy requirements lowered for ourselves is through the use of enzymes, which were proteins that we mentioned a little bit earlier. So an enzyme is a protein which helps catalyze a reaction. It does not cause the reaction to occur, nor does it actually affect the products or reactants. It simply steps in and helps reduce the energy of activation for that reaction. One other nice benefit of the enzyme is it actually helps increase the speed at which the reaction occurs. So it lowers the energy cost of making the reaction happen, and it helps it occur at a faster rate. Again, it does not actually change the products or the reactants, however. It's just a protein, it steps in, it basically makes things a little bit easier, reduces the energy cost, makes it happen a little bit faster, more efficient, and then backs off. So really, really handy. And almost all of our body's major biochemical reactions are governed by enzymes. And enzymes can vary in degree of how much enzyme is available or how active they are within the cell or any given part of the body. Now, we also know that each enzyme has an optimal range of both temperature and pH. And when they fall out of that range, they can degrade or simply not be as active and function as well as they could. So we see a lot of different examples of this in the muscle tissue where we can see, especially with pH, the acidity of the environment might actually cause an enzyme to become activated or deactivated depending on what the optimal range is for that particular enzyme. We can also see similar effects with temperature. So we know that enzymes are very, very helpful. They're proteins. They assist in almost all chemical reactions in the body, but they do have some constrictions. They can't always be super, super active, and sometimes we actually have to take steps to deactivate certain enzymes to prevent certain processes from happening, which is also pretty cool, right? So we get positive and negative feedback. Let's go on to the next slide. So here's an example of what this just looks like, right? If we were looking at energy requirements for a reaction. So that red line was an example of a reaction without the enzyme. So we can see there's a really, really high activation energy required for this. However, that blue line shows the difference of the same reaction now being performed with an enzyme. And we can see a very, very significant reduction in the amount of energy required to put into that system to get that product, which in this case we had carbon dioxide and water, and then we got bicarbonate as a result, right? So again, just meant to illustrate, it helps reduce the energy cost for a lot of these reactions to occur. So let's go on to the next one. So here we can see one of the pivotal, most important things that we can talk about for today. Any ideas what this picture is? ATP. adenosine triphosphate. Now, what we have here is an adenine nucleotide, a ribose, which is a carbohydrate backbone, and then we have 3 phosphate groups, right? So again, we see that carbohydrate carbon skeleton kind of holding these things together. And then what's of note here are these 3 phosphate groups. These are really, really, really high energy bonds that are held together. And what we find is that when we break these phosphate groups off of each other, they release quite a bit of free energy. Well, it turns out ATP is going to be the usable energy form for all of our biological purposes. So everything that requires energy generally needs to form ATP and then break ATP down. So it's the usable form of energy within the body, right? Even when we talk about things like carbohydrates, proteins, fats for energy purposes, ultimately they are being used to resynthesize lost ATPs. ATP is the usable chemical form of energy in the body. So again, I think this is something that a lot of my students tend to miss a little bit, where they just think carbohydrate leads to kilojoules, right? Well, really, carbohydrate is being used to resynthesize ATP, which then leads to kilojoules, right? So it's kind of another step along the way. So again, this is the source of all energy within the body for our purposes, and that energy comes in these high energy phosphate bonds, right? That's a lot of potential energy being stored there, and once they're broken apart into ADP and inorganic phosphate, it releases a lot of free energy, and that's the energy that we try to tap into to perform exercise and sport, right? So again, this is kind of the big one here, and again, we can see why understanding a little bit of chemistry is probably worthwhile, and that's why we kind of are doing this lecture. So ATP, source of energy for all of our biochemical reactions. It comes from breaking off those phosphates from that ribose and adenine structure. Okay, let's go on to the next one. All right, so we know again that the breaking of those chemical bonds provides the energy for all of life's various functions. Now, typically when we're talking about bioenergetics and exercise physiology, we're trying to figure out how we resynthesize ATP through a couple major processes. The first one is through phosphocreatine breakdown. So we actually store phosphocreatine in our muscle cells. When our ATP stores run out during a high intensity exercise, they tend to run out really, really fast within a couple seconds. CP is right there. It gets broken down and helps resynthesize lost ATPs, and it also helps buffer like hydrogen ion accumulation, which is really important. We're also talking about the reformation of ATP through glycolysis, which is through carbohydrate breakdown, as well as through oxidation, which can include carbohydrates, fats, and proteins. It's a much longer, drawn-out process that you'll learn more about later on in exercise physiology. Okay, so now that we have a pretty good grasp on some basic chemistry, some basic understanding of biomolecules and some basic chemical reactions, now we can move into some basics on cell physiology. So here on the slide, you can see the structure of a cell. And I really like this diagram. I don't even remember where I got it from, but I think it's really cool. Maybe it's just because it's colorful and I have ADD and I like the colors and the pictures. Whatever, it's still really good, right? So we can see the cell is this very, very complex machine-like structure with all sorts of different organelles, all of which have their own kind of structure and function. So we're not going to go through all of these things, but what we are going to do is talk about a couple really important ones that you should know going forward so we can have a better understanding of exercise physiology later on. So first off, we know that the cell has a number of different organelles. The first one that we see is that big outer layer, what we call the cell or plasma membrane. And this is a semi-permeable membrane that separates the cell from the external environment. And again, that membrane is composed usually of some type of lipid layer, whether it's a bilayer or single layer. Usually the outer membrane is typically formed with lipids, and you can kind of think of it as kind of like a net-like substance. So it helps keep things in, but it is semi-permeable. So small things can kind of pass in and out somewhat freely, whereas larger things can get caught in the net and have to be actually released into the cell through specific mechanisms. So the cellular membrane helps enclose the cell. It regulates things in and out of the cell. So some things can pass, some things cannot, and some things actually need to be brought in and out of the cell very directly. And it is semi-permeable, right? So some things can come, some things can go, others cannot. Within that, we have the cytoplasm, which is the fluid portion of the cell, and it contains all of the organelles. So it's kind of like hard to describe, but it's kind of like the jelly-like substance really within the cell that just kind of holds everything together. It's kind of like, for lack of a better term, glue, although it's not really like glue because it's very fluid and things can pass through it very easily. So the cytoplasm contains all of the organelles of the cell. It also has parts of the cytoskeleton. There are structural compartments of this cell itself with different molecules like actin and kinesin, which can move things throughout the cell, right? It also contains some of the enzymes involved in some biochemical process like glycolysis. So some of those things require certain organelles, some of them don't. And in the example of breaking down carbohydrates for energy, a lot of that actually occurs right there in the cytoplasm of the cell itself, which is pretty neat. It doesn't necessarily have to be shuttled off into a specific organelle, so it makes it really quick and easy. All right, so the cytoplasm, the fluid portion of the cell containing all the organelles, right? We have a couple other things that are noteworthy. First, we have the smooth endoplasmic reticulum, and if you want to go back and backtrack and take a look at the picture again, that might be helpful. The smooth endoplasmic reticulum is a part of the cell that is involved in lipid production and metabolism and can kind of help clean some of the crap out of the cell and keep it from getting too like toxic and gross in there. Not as important for our discussion. What is important is the rough endoplasmic reticulum, which is an area of the cell, and you'll see it's the one that has kind of the waves and the little bumps and dots all over it. Those little dots are called ribosomes, and that is the site where different proteins are actually packaged and made, which is really, really neat. So ribosomes from the endoplasmic reticulum link different amino acids together to form proteins that are dictated by things like messenger RNA. Sometimes you'll see mRNA, that stands for messenger RNA. So the mRNA is basically kind of like a recipe that comes up to the ribosome. It's like a cookbook and it says, hey, I want this type of pizza or this cake. The ribosome says, no problem, puts all the ingredients together and forms big protein chains, right? So in this case, we're talking about amino acids and proteins. Those proteins can be used to signal all sorts of crazy stuff within the body or within the cell itself, or it could be used for all sorts of different tasks, right? So mRNA tells the ribosome what to make, the ribosome puts them together. Really, really easy. So in the muscle cells, we have another kind of unique part of the endoplasmic reticulum. It's a section of smooth endoplasmic reticulum that we call the sarcoplasm. So anytime you see the prefix sarco, that usually means muscle, right? So we said cytoplasm. In this case, we have sarcoplasm, which is a little bit different. And for us, it's actually the site where we actually store and release different ions, particularly calcium ions that are used for muscle contraction. So in the muscle cell, we have that the sarcoplasm, which is responsible for maintenance of ion concentration across certain membranes within the cell. So it helps spill out calcium and pull it back. Really, really easy. All right, the next one. Kind of the weird, gross, booger-looking thing in the cell is what we like to call the Golgi apparatus. And it's really, really funny, and it's usually very distinct when you look at a picture of the cell because it kind of has that snot-like shape. So the Golgi apparatus of the cell is kind of a unique structure, which kind of, what I like to say, functions like a post office in the cell. So within the cell, we can package all sorts of stuff to be used for transport across the cell. And most commonly, we're talking about proteins because they signal for other things, but it can include all sorts of stuff. So typically, when something needs to move across the cell, it's enclosed in a vesicle, which is kind of a spherical membrane that envelops whatever it is that needs to be transported. That usually gets sent off to this Golgi apparatus, where the Golgi apparatus will kind of envelop these vesicles, so you can kind of see it's like a booger snot looking thing, and it basically moves into a bigger booger snot looking thing, which is kind of gross, but it basically envelops or invaginates the vesicle, takes the contents out, reprocesses it, and then we'll send it off to wherever it needs to go. So it very much functions like the post office in the cell, where we have certain things like proteins that need to get sent to different areas, whether they're in the cell or out of the cell. The Golgi apparatus ensures that things are going where they need to be, and it repackages them in little vesicles, where basically, and it's really actually cool if you ever see a video of this happening, basically it kind of pinches off the vesicle right off of the Golgi apparatus, so it will kind of form a membrane around whatever particular item needs to be shuttled off, makes that kind of a spherical shape, and then just kind of pinches it off and then sends it off to other various parts of the cell, or possibly even outside of the cell. So it's really, really neat. It gets contents from inside the cell and from outside the cell, and then repackages them, takes them where they need to go. Make sense? Pretty simple. Now, one of the ones that's probably the most important for our discussion of the human body within the cell is the mitochondria. Now, the mitochondria is the site for cellular respiration. This is where all of the oxidation of fats, carbohydrates, proteins, all that stuff will occur. So when we need to break those things down for energy purposes, catabolism, right? They have to make their way, one way, shape, or form, into the mitochondria to undergo a long series of chemical reactions to break those things down. So this is the site where we see the electron transport chain, the TCA cycle, oxidative phosphorylation, all that stuff. Now, you'll learn more about those things later on in subsequent videos. For now, we'll just kind of want to call attention to it. So again, the mitochondria, and if you're looking at pictures of a cell, it's usually a very dark kind of ellipsoid kind of shaped area within the cell. And again, this is kind of what most people will call the powerhouse of the cell, simply because this is where lots and lots of energy are produced. Now, we know that the number of mitochondria within a cell can vary depending on the person's genetics or trained state. It can be more densely packed or less densely packed. Either way, we know that that mitochondria is going to help take things like carbohydrates, fats, and proteins, break them down into their usable chemical energy form. Unfortunately for us, getting them to the mitochondria is kind of a pain in the butt because, for example, in the example of fats and lipids, we have to get them from the fat storage site. So for example, like from the gut, they got to break down into a fatty acid form, make their way into the bloodstream, make their way into the target muscle cell, cross that barrier, get into the sarcoplasm, make their way to the mitochondria, cross into that barrier, and then undergo a number of different chemical reactions. So unfortunately, this is really, really slow and very, very logistical. So it doesn't happen very rapidly and very readily during exercise. But at rest, it works very, very well. All right, let's go on to the next one. So certainly last and not least in this discussion is the nucleus of the cell. Most people know the nucleus is the site where the DNA is contained, and that is absolutely correct. And that is very, very important for processes like mitosis and meiosis, which is cellular replication, transcription and translation of proteins, and of course, protein synthesis. Now, one thing we want to establish, and we tell our students this all the time, when you hear the phrase protein synthesis, a lot of times people just think skeletal muscle, right? Getting more jacked. Yes, that's part of it. But again, proteins actually regulate and signal all sorts of different cellular functions and actions. So we know that protein synthesis is kind of the governing, organizing factor for all of our major biochemical processes. So when we see proteins out in the blood, they're usually signals that something needs to change or something needs to occur in order to maintain homeostasis, right? Which is very important. So we want to think about the DNA of the cell and protein synthesis, not just as skeletal muscle protein synthesis, but protein synthesis for the entire system, governing all major processes, which is very, very important. So just because you hear protein synthesis doesn't necessarily mean just skeletal muscle. It can include all biological functions. The nucleus is what contains all that information, right? So everything that makes you is located in there. And what's really, really neat about exercise and nutrition and recovery and all these different topics is when you do those things and when conditions are good, you actually are changing the way your DNA is expressed phenotypically, right? So you kind of have some preset things in your DNA that's already making you, but you can change some of those things along the way, which is pretty cool through diet and exercise. So definitely worth noting that all of your genetic information is stored right there in the nucleus and can be changed a little bit here and there based on what you do. Pretty cool. All right, so here's just kind of a quick description of this. Now I've taken entire classes as a biochem major back in the day about DNA replication. This is just kind of the short, quick, dirty version where we can see there is a need for a specific protein, right? We start transcribing. DNA results in some type of mRNA blueprint, right? Kind of that recipe. mRNA leaves the cell, goes to the ribosome. Ribosome takes the amino acids, put proteins together. Proteins get sent out to do whatever they need to do, right? That's like about as crude of a description of this as it gets, but probably enough to kind of get you started for now. This is actually a super, super vast, crazy, complex machinery of things going on. Really, really, really fun to study. So if you've never actually checked out how DNA is replicated before or how proteins are made, check it out. It's really, really neat. This doesn't really do it total justice, but will at least get you warmed up for these concepts later on. All right, let's go on to the next one. So within the cell, generally, we already mentioned things are packaged in vesicles, right? Whether it's debris or ions or proteins that need to get moved around, we have this kind of fluid enclosed membrane that grabs whatever it is, it envelops it, and then we usually can send it off to different parts in the cell. Now, typically, this is done through the processes of what we call endocytosis, which is the uptake of materials by enveloping the membrane, right? So again, we have kind of booger going into a bigger booger, where the bigger booger sort of envelops the small one and they become one. That's endocytosis. Exocytosis is an active transport mechanism, which we'll talk about in a second, of actually expelling material. So this is when we have stuff that's inside the big booger and we're kind of pinching it off into a small booger to be sent off wherever it needs to go, right? So endocytosis is enveloping, right? Exocytosis is expelling material outside of usually the Golgi apparatus, but other other organelles as well. Now, once we have formed a vesicle and we have some item that we're trying to move, we typically move them across microtubules within the cell. So the microtubules I like to think of are the infrastructure of the cell. These are proteins that kind of make an interconnected, lack of a better term, kind of highway where things can move about the cell from front to back, left to right, up and down. So usually we have things like actin and kinesin, which use electrically charged polar ends to guide things across the cell. So when something needs to move, we can have a vesicle, and it will essentially walk along some of these cytoskeletal tracks to wherever it needs to go in the cell to drop off whatever it's carrying. This is something that's really, really neat. If you can look at some 3D images of things moving up and down kinesin in the cell, really, really fun and really, really cool. So again, we kind of have some infrastructure in the cell. We have some proteins that guide things across the cell, and typically what we see is these vesicles kind of walk along these infrastructural proteins and guide these items to wherever they need to go, right? Okay, moving into the next one. Moving things in the cell, not a huge deal, right? We can create vesicles, we can kind of move them around. A lot of things can actually move around through passive means. If we need to move something in and out of the cell, it's a little bit more complicated, a little bit more challenging. Now, this can happen through a number of different mechanisms. The first one is a group of mechanisms we just call passive transport. Passive transport essentially means the passing of materials without putting in any significant amount of energy. Basically, it just happens freely, no energy is put in. So the most common example of this is what we call diffusion, which is where small molecules essentially can pass through holes in a membrane when there is a concentration gradient between two things, right? Now, concentration gradient basically says we have areas of high concentration in one area, an area of low concentration in another area, and what diffusion does is attempts to equilibrate these two things so that their concentrations are roughly the same. This can happen across semi-permeable membranes with small molecules, right? So essentially with diffusion, we have something going from high to low to equilibrate the two. And this happens all the time, very, very simple. We can also see what we call facilitated diffusion, where we actually have carrier proteins that are located on the membrane surface that are targeting specific either molecules, ions, or different compounds very, very specifically. And once those things kind of make contact with the protein, it will allow them to pass either in and out of the cell more freely, right? So it's not quite a free passing in and out. It's more of like a baseball glove detecting a baseball, and that says, oh, okay, that's what's supposed to be here. We'll bring it into the cell. No big deal, right? Other things might rely more on diffusion. Some things like ions or really, really big biomolecules like fats often require carrier proteins to be brought in. It's kind of like a big gate. And then last on this list is osmosis. This is just a really, really specific example of diffusion specific to water, where water basically kind of moves from areas of high concentration to low concentration. It's a little bit more specific in terms of the actual concentration is measured as osmotic pressure, which is very similar to concentration, not quite the same, but for our purposes acts basically the same as diffusion. In this case, we're just talking about water. So if you see the word osmosis, almost synonymous with diffusion specifically to water. Very, very simple. So that's passive transport. Again, things can just kind of move from areas of high concentration to low concentration without really putting any energy into it. On the other hand, we have active transport, which is another part of cellular movement. This, as the name implies, requires energy. You have to put energy into this to actually get it to move. So what's really neat about active transport is that it can go against concentration gradients. So for passive transport, we said there has to be a concentration gradient and it has to go from high to low. In active transport, we can actually go against these high to low concentration gradients, especially for things like nerve conduction and muscle contraction, where we can actually go against it and make the concentration gradient even bigger, which is cool, but it requires energy. For some things that are too big, like glucose or fat molecules, sometimes they're just too large to pass through the cell membrane. They actually require some type of protein gate channel to actually move in, which requires energy to be put into that system to allow them to pass through. They usually require also some type of electrochemical gradient. So we said usually a concentration gradient was required for passive transport. For active transport, generally we need some type of electrical gradient as well. It means there has to be kind of a voltage or charge difference across a couple membranes. And then last but not least, we can use the same processes of endo and exocytosis to either allow things into the cell and form vesicles, or we can pop a vesicle out and kind of let it exit the cell and move into another cell. So these are the major ways that things are kind of moved within the cell or moved out of the cell. So last on my list of things that we're going to talk about for today is just a quick kind of overview here. We can see muscle cells, right? So the first one we looked at was just kind of a typical, excuse me, what they call eukaryotic, for what our purposes we'll call human cell, right? This is more of a specific example of that, and that is of skeletal muscle, right? And here we can see some of those same organelles, right? There's the nucleus, we can see mitochondria, we can see sarcoplasmic reticulum in there, all sorts of good stuff, right? But in this case, we also have the cellular machinery involved in actually generating tension inside that cell. And so you're going to learn more and more about this in the exercise physiology lectures, but it's just here to show you that they are slightly different. So if we're talking about a muscle cell, it might not necessarily look at the same as the cell that we saw on the earlier slide, right? It's going to contain a lot more contractile material, which is really, really fun and really, really cool to learn about. But many of the same, virtually all the same organelles are still in there as well. So again, the muscle cell, very, very important differentiation is its sole purpose is to generate tension and force, right? So we're going to actually see really, really, really in-depth contractile material in there. And this might vary a little bit from cell to cell. So we'll see like type 1 fibers might have a little bit more vascularization. They might have more mitochondria, whereas type 2X fibers might have a little bit less packing density of those things, but more and more and more actin and myosin filaments to create bigger forces. So it's just worth noting, same idea of the same organelles, same cell stuff, now just placed in the context of skeletal muscle. All right, hopefully that gets you up to speed on some pretty basic ideas. Stay tuned, we'll have some more expanding ideas on basic cellular physiology in the next video. RPU, RP+, thanks for tuning in. I'll catch you next time.